Hello again, and welcome to the Chapter 15 online lecture. You should use the information in this lecture to complete the Chapter 15 guided notes, which of course you should complete before you come to class. In this chapter, we're going to discuss what's known as the chromosomal theory of inheritance. This theory helps to explain how the specific chromosomes that carry certain genes, their specific locations on those chromosomes, which if you remember are referred to as a locus or loci plural, and even the shape and size of the chromosomes themselves can affect how genes are inherited. Now remember that Mendel didn't know anything about DNA or chromosomes at all, but nonetheless he was able to determine the basic patterns of inheritance simply through observation and logic. That's pretty impressive. Now some of his ideas were pretty spot on, but others were a bit off. Our current knowledge about chromosomes actually helps us reinforce some of his ideas and expand on others. Mendel described two laws that he called the law of independent assortment and the law of segregation. These laws help describe how traits are passed from parent to offspring in random combinations. Today, we know that this is mostly true because of how chromosomes behave during meiosis. Remember that during meiosis, the maternal and paternal chromosomes get all mixed up together through crossing over, and then separate from one another through two separate divisions. Now, because of this knowledge, you know that when your body makes egg cells or sperm cells, those cells are going to contain one copy of each chromosome, so 23 total, but there is no way of knowing which of your maternal or paternal genes each of those chromosomes carries. Now, there are exceptions to Mendel's rules. It turns out that some genes tend to travel together on chromosomes from parent to offspring and are unlikely to become separated by crossing over. They tend to stick together. We refer to these types of genes as being linked. None of the traits that Mendel studied happened to be linked, so he didn't know anything about linkage. But linkage can affect genetic outcomes, so we need to discuss it. Linked genes tend to travel together from parent to offspring and are not likely to be separated by crossing over. For instance, consider a pea plant with the genotype big P, big L, little l. In other words, it's heterozygous for two different traits. Now, if we looked inside the pollen grains or the egg cells produced by this plant, we would expect to find 1/4 big P, big L, 1/4 big P, little l, 1/4 little p, big L, and 1/4 containing little p, little l, according to Mendel's rules. However, in nature, these types of plants mostly produce the gametes containing big P, big L and little p, little l. Why is that? Well, this occurs because the genes for characteristics P and L are linked together. They're located on the same chromosome and they're located pretty close together. So genes that are located on the same chromosome are linked to one another. And the closer together the loci of the two genes, the more linkage exists. Genes that are located on different chromosomes, however, are not linked, and they separate randomly from one another. So the only genes that behave by Mendel's rules are those that are located on separate chromosomes and are not linked. As an example, let's say that we take two pea plants, both of whom are heterozygous for the P and L traits, and we breed them together and we look at the F1 offspring. Now in this example, the P's represent flower color. So big P represents purple flowers, while little p's represent red flowers. The L's represent the shape of the pollen grain. So in pea plants, there are long pollen grains and there are short pollen grains. Big L represents long, little l represents the short pollen grain. Now, according to Mendel's rules, this cross should produce a 9:3 to 3:1 phenotypic ratio among the F1s. In other words, we would expect that 9 out of 16 of the offspring are going to be purple with long pollen grains, that 3 out of 16 will be purple with short pollen grains, that 3 out of every 16 are red with long pollen grains, and that only 1 out of 16 are red with short pollen grains. However, in nature, this cross mainly produces these two phenotypes, purple with long pollen grains and red with short pollen grains. So we get a lot more of the red with short pollen grains than expected. Why is this? Well, this occurs because the traits for flower color and for pollen shape are not dispersed randomly among the sex cells as expected. They don't follow Mendelian rules because these two traits are linked. They are located on the same chromosome and are located close together on that chromosome. Here we have a homologous pair of chromosomes that carry the traits for flower color and for pollen shape. This chromosome carries the allele for purple flowers in combination with the allele for long pollen grains, while this other one carries the allele for red flowers in combination with the allele for short pollen grains. Here we have the crossing over event, which of course occurs in prophase 1 of meiosis 1. Now notice that these genes for flower color and for pollen shape are located really close together on this chromosome. Because they are located really close together, it's very unlikely that crossing over will occur between them. So because these guys are already close together, they tend to stay close together, and crossing over has actually happened over here. Crossing over isn't separating these alleles from one another. Here we have the chromosomes after crossing over is complete. Notice that the big P's and big L's tended to stay together. They didn't get separated or mixed up with the little p's or little l's. And the little p's and little l's tended to stay together as well, even though other genes on this chromosome did get mixed up into new combinations. Here we have the 4 separate chromosomes that result from the end of meiosis II. These 4 chromosomes will go into 4 separate sex cells. Notice that 50% of these chromosomes contain big P and big L in combination, and the other 50% contain little p and little l in combination. Because 50% contain big P, big L, and 50% contain little p, little l, when two heterozygous plants carrying these gametes are bred together, the offspring are mainly going to be purple with long pollen grains and red with short pollen grains.